So now let's go to the second quiz. So this is a very basic question, and you know that the DNA instructions are useless if they are not transcribed and translate. And I'm asking you in particular, what is the name of the process in which you make a copy of the RNA using as a template, or you make an RNA copy using as a template the DNA? And the answer is very easy. It's called, as we will show later, transcription. So this is very easy. So if you have doubts, you can go to the video lecture. But transcription is the synthesis of RNA under the direction of DNA, and then after the transcription, translation takes place, and in eukaryotic this happens in the cytoplasm. Well, in the prokaryotes, the transcription takes place in the beginning. The copy is made in the nucleus, and then the messenger RNA goes outside where it's translated in the cytoplasm. So definitely transcription. Mutation is a change in the DNA, so no. DNA replication is when you have a copy, but when you make a copy of a DNA. So not an A, definitely wrong, because it's when you use the RNA to the messenger RNA and you create proteins using not only this RNA plus tRNA and ribosomal RNA. Another type of RNA, but in this class we will focus mainly on these three. And again, this question is asking you about the transcription, and as we said before, the transcription is the synthesis of RNA. So again, synthesis of RNA, this is the correct one. Here we just talk about it. The replication of DNA is not transcription. This is the copy of DNA, it will be replication. Metabolism of sugar is not related at all. Synthesis of proteins is the translation, and synthesis of DNA will be replication. So E is the only answer. Okay, this one is also a very basic question, and I'm asking you what is an anti-codon. So please read this, and in the next section, in the next slide, I will show you a schematic to make it clear. So I'm sure that all of you know that the answer is B, that an anticodon contains a sequence of complementary to the codon. So here, if you see this schematic, you have the RNA, the anticodon loop, and here you have the messenger RNA. So two type of RNA, and the anticodon is in the tRNA, and the codon is in the messenger RNA, and so they are complementary to each other, and this complementary allow them to attach to each other to later. Here is where, if you remember, the amino acids will be here. So definitely is the correct one. Contains the sequence same to the codon. So if you see here, the anticodon is the complementary, so this is wrong. If it was the same sequence, they will not attach to each other. Contains the base thymine. This is this is not an answer, and D is wrong. Okay, tRNA is involved in. So previously I asked you about the anticodon, and I talked about the messenger RNA, and so now I want to remind you what is the function of the transfer RNA. So here, as we said before, we focused last time in the anticodon and the codon, and now we are going to move into this region. That is where you find, as I said before, the amino acid. So the tRNA is involved in carrying the amino acid, and then, as I said before, during translation this amino acid is released to form the protein. And definitely B is incorrect. It's not carrying a ribosome, it's not carrying a glucose, and it's not carrying lipids. So the only option is amino acids. Okay, this one is also very easy. Amino acids are joined together in order to form. Here we already studied this, so you know that the only answer is proteins, that the monomers of protein is amino acids. Well, the other ones definitely are wrong. Yeah, so if you remember, to make proteins, you take amino acid, you join that through peptide links, and then polypeptides join together form proteins. So the only answer is C, and the other ones are wrong. Again, this is a very easy question. Here I'm asking you about the 3 bases of the messenger RNA. So the only option here is a codon. We already studied the anti-codon here, but we know that the codon will be the one for the messenger RNA. And in case that you have any doubt about this, I recommend you to go to the video lecture, to this section. Here I talk about what is a codon, and I show you this structure, and I talk about that the codons are form of messenger RNA, and they are read from a 5' to 3' direction. I also talk that the codons are triplets. So here again, remember, they are form of messenger RNA. So the right answer, as we say, is codon. This will be, because it's messenger RNA, this one will be tRNA. So this is wrong. Triplet, they already told us that they have 3 bases, so this is a trait of a codon, but it's just wrong. And a gene is formed mainly of DNA, and it's not a triplet, so this is just wrong. So the only option is C. Okay, so this is a very easy question that maybe you already know the answer. RNA use U instead of T, and this is a difference between RNA and DNA. So the other options just don't make sense. So B is the right answer. And if you have doubt about this, please go and study the difference between DNA and RNA. That you're just we already studied that before. So go and look into that in the previous video lecture, but I'm sure that you already have this clear. Okay, this is a very simple question, and I gave you this DNA sequence, and I said that there is a part of a gene, and I ask you how many amino acids are called by this first code by this message. So just a clue, remember that there are codons and there are triplets, so you can count the number of bases, and in this way you will find the answer. So if you count it, here you know that there is 1 triplet, 2 triplets, 3 triplets, 4 triplets. So the answer is 4, and it's codon. Here are the codons, because these go for DNA to mRNA, and this mRNA will be the codons. So easy, the other options don't make any sense. Okay, now another easy question. Translation is crucial to the process of making proteins. Which of the statements base describe what takes place during translation? So give a read to the answers, and in the next question, in the next slide, I will give you the answer. So you can go to the video lecture again or go to your textbook, and you know that the translation required first a messenger RNA that goes outside of the nucleus, and outside of the nucleus in the ribosomes, this messenger RNA is read, and there is a synthesis of polypeptide. So you turn the information of the messenger RNA into amino acids. So for that reason, B is the right answer, while a copy of a chromosomal DNA is created. This will be more replication. C, a copy or an RNA copy of a DNA strand is made. This is more transcription. And D, okay, so this is wrong, and the instructions from a DNA in the nucleus are brought to the cytoplasm. This is just not a complete answer. So the best answer is B.